So, good morning everyone. In this video season, we will discuss the another very important topic is Kingdom Protista. So, before we will discuss the Kingdom Monira. So, generally, R.H. Whittaker proposed a 5 kingdom classification. R.H. Whittaker proposed 5 kingdom classification. That is Monira, Protista, Fungi, Plantae and Animalia. So R.H. Whittaker proposed 5 kingdom classifications. We are already finished one kingdom. That is Monira. We are already finished that. Now we are going to discuss the Kingdom Protista. Now we are going to discuss the another very important topic is Kingdom Protista. So in this kingdom, all eukaryotic organism present under the kingdom is Protista. All eukaryotic organism present under the kingdom is Protista. All single cell eukaryotic organisms. All single cell eukaryotic organism present under the kingdom is Protista. So boundaries are not well defined in this kingdom. So boundaries means there is no infinities. So the number of organisms are included in this kingdom. So that's why what happened, the boundaries are not well defined in this kingdom. So in this kingdom, they are having actually single cell organisms and multi-cell organisms. So what about actually single cell organisms? Single cell organisms means the organisms it is consists of only one cell. That is known as actually single cell organisms. So some other organisms is consists of more than one cells. That is known as multicellular organisms. So now what is the main concept of the Kingdom Protista? So in this kingdom, all single-celled eukaryotic organism present under the kingdom is Protista. Boundaries are not well defined. Boundaries are well defined in this kingdom. So the boundaries are very well defined in this kingdom, and as usual, nucleus also very well developed. Nucleus and nucleus membrane also very well developed in this kingdom. So these kingdoms. So these kingdoms are generally included in single-celled eukaryotic organisms. So these kingdoms are generally included in single-celled eukaryotic organisms and well-defined nucleus. So means in this kingdom it is consists of well-defined nucleus and other membrane-bound organelles. Okay. So and this kingdom it is consists of a flagellus. This kingdom it is consists of a flagellus and as usual in cilias. So flagella and cilia, they are mostly helps in actually motility purpose. They are mostly helps in actually motility purpose. So here cilia also be there and flagella also be there. So the cilia are mostly present in actually paramecium and flagella are mostly present in actually euglenoids. Okay. Now, so in this kingdom, it's again classified into 5 types. Means the included members in the kingdom is, so which type of organisms are included under the Protista? So diatoms, chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. There are 5 members are included in kingdom Protista. So what are those? What are sub kingdoms in the Protistas? So Kingdom Protista again classified into 5 types. Kingdom Protista again classified into 5 types. That is chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. There are 5 types in Kingdom Protista. So these Kingdom Protistas are generally linked with. So most of the Protista membranes are generally, they are primitive aquatic organisms. They are primitively aquatic organisms. Most of the Protistants, they are primitive aquatic organisms. So why? Because they are mostly present in water. So that's why we are saying that it is a primitive water, means a primitive aquatic organisms. So these kingdoms are generally linked with plants, animals and fungus. These Protistants are generally linked with plant and as usual in animals and other in fungus. Now, a reproduction of Protistas. So, what is the main concept of reproductions of Protistants? So, here the three types of reproductions are mostly occurs in Protistas. So, the main thing is vegetative reproductions occur to the fragmentations, asexual reproductions occurs to the fusions and spores. Asexual reproductions are mainly occurs through the fusions and spores. And as well, sexual reproductions are occurs to the formation of gametes. Formation of gametes, that is fusion and fusions. And both two gametes, the form of zygote, zygote undergoes as usual meiotic divisions, then form of another new haploid organisms. Next. So, right now we have discussed kingdom again classified into five types. Kingdom again classified into five types. They are kingdom Protista. Kingdom Protista again classified into five types. That is chrysophytes, dinoflagellates, euglenites, slime molds and protozoans. Now we have first discussed the chrysophytes. So what about actually chrysophytes? So the chrysophyte members are mostly present in freshwater and as usual in marine water. So they are mostly present in actually these chrysophyte members. There are number of chrysophytes members are there. So these members of chrysophytes, they are mostly present in freshwater and as usual in marine water and marine habitats. So some other chrysophytes, they are microscopic, means it is very small size of organisms are there, that is known as actually microscopic, and generally they are float on the surface of water. These microscopic organisms are generally float on the surface of water in stagnant of water. So they are generally float on the surface of water in the water currents like that. Plankton is the best example of microscopic organisms. Most of the chrysophyte members, they are actually photosynthetic organisms. Most of the chrysophyte members generally they are photosynthetic organisms. So this chrysophytes is also known as actually diatoms, desmids and golden algae. The chrysophytes is also known as actually diatoms, desmids and golden algae. So these chrysophytes, so the chrysophyte it is consists of actually two thin overlapping cells. The chrysophyte it is consists of actually two thin overlapping cells. Those cell walls are generally made up of siliceous of cell wall. Means that silica granules are present in these chrysophyte cell walls. So those two thin overlapping cells, both are fit together a soapbox like structures. Both are fit together a soapbox like structures. Here the beautiful images are there. See once. So both two walls are generally made up of actually silica granules or silica crystals. Okay. And both are fit together a soapbox like structures. Both are fit together a soapbox like structures. And these diatoms are chief producer in the oceans. Chief producer in oceans. So they have this siliceous cell wall. Two thin overlapping cells, both are fit together as soapbox like structures. Okay, and they are chief producer in the oceans. Right now we are already discussed. So the cell wall deposit due to the diatoms over billions of years in their habitats. Means over the cell wall is due to the depositions of billions of years in their accumulations, such is known as actually diatomaceous earth, and it is also known as Kishal Guhar. It is also known as actually Kishal Guhar. So it is also known as actually Kishal Guhar. So what about actually Kishal Ghose, diatoms left behind the large amount of cell wall due to the depositions on the earth. That earth is known as actually means that accumulation of diatomaceous cell walls, that is known as actually diatomaceous earth. So it is also known as Kishal Ghose. So this Kishal Ghose are mostly helps in actually. So this Kishal Ghose are mostly helps in actually filtration of oils, syrups, and polishings. So the diatoms it consists of actually two flagellas based on the structural symmetries. Okay, diatoms it is consist of a two flagellas. Both two flagellas are mainly based on structural symmetries. So one is central diatoms are present in actually radial symmetricals. So they are having actually right now we are already discussed it is consist of a two symmetrical structures. One is central diatoms are generally radial symmetricals and pinnals are generally bilateral symmetrical. So these diatoms are mostly consist of two types of symmetrical structures. So one is radial symmetricals, another one is bilateral symmetrical. Radial symmetricals is mostly involved in centrals, in bilateral symmetricals are involved in pennants. So they produce actually asexually by the binary fusion in diatoms. So asexual reproductions are mostly occurs to the binary fusions. Okay, and sexually reproduction and the formation of gamete. Okay, right now it is a finish. Now here one important is there. As a sexual reproduction result, the formation of one spores. That spore is known as actually rejuvenatory spores. It is very important point. This question will be asking in actually 2018 NEET. Okay, where the rejuvenatory spores are found in diatoms or chrysophytes. Asexual reproductions occurs to the spores or binary fusion. Next, sexual reproductions or formation. These sexual reproductions occurs through the rejuvenatory spores. Now, what is exactly meaning of rejuvenatory spores? Rejuvenatory spores means both two gametes are formed and formation of zygote is a diploid in condition. Both two gametes are generally haploid in condition. Zygotes are generally diploid in condition. This zygotes undergoes meiosis and it is produce haploid spores. So this haploid spores and this gives rise to young spores. Means zygote is gives rise to young spores or young new spores, that is known as actually rejuvenatory spores. Next thing. Now we are goes to the another one, dinoflagellates. So what about actually dinoflagellates? These dinoflagellates are generally marine algae. Dinoflagellates are mostly present in actually marine algae. So this dinoflagellate it is consist of many colors. What are those? It's having actually colors. They are having the yellow color, green color, blue color, and red color. So these colors is mainly depends on pigment. These colors is mainly depends on entire pigments. So means this dinoflagellates is already consist of a pigments. Okay. So this pigment is gives rise to the colors. And this dinoflagellates. This dinoflagellate it is consist of a two cell walls. This dinoflagellate it is consist of a cell walls. Third cell wall is generally made up of cellulose or stiff cellulosic plate. Third cell wall is generally made up of stiff cellulosic plate. Next, so the flagella. The dinoflagellate is consist of a two flagellas. The flagella spinning movements inside of the flagella spinning movements is inside of the wall plates. Okay, the flagellas are farrow. The flagellas are generally they are having actually two flagellas. One flagellas are arranged in longitudinally, another flagella is transversely. So it is consist of actually two flagellas. These two flagellas are farrow between the walls. These two flagellas are farrow in between the walls. Next thing. So this flagella, spinning movements are occurs. The spinning movements are occurs inner side of the wall plates. The flagella spinning movements are occurs inner side of the wall plates, such is known as actually whirling whips. So it is a specific characters in dinoflagellates. Whirling whips are mostly present in only dinoflagellates. Next, this dinoflagellate, it is consist of it is condensed nucleus. This nucleus, it is also having actually chromosomes. It is also having actually chromosomes. And even in interface, it is does not contain actually histone proteins. Here the one main important point is there. So histone proteins does not contain, means histone proteins are completely absent in dinoflagellates, such is known as actually mesokaryote. It is known as actually mesokaryote. Some marine dinoflagellate, so like Noctiluca, some marine dinoflagellate like Noctiluca, it shows the bioluminescence. Now what about actually bioluminescence? Some insects in night time, they exposing light. Some insects in night time, they expose the light. That is known as actually bioluminescence. Some other marine dinoflagellate shows actually bioluminescence. The best example is Noctiluca. And next thing. So we are already discussed. They consist of two flagellas. One flagellus are arranged in transversely, another flagellus are arranged in longitudinally. These two flagellas are there now. And here red dinoflagellates. Like that, example is red dinoflagellate, example is Goniaulux. So this Goniaulux undergoes rapid multiplications. Goniaulux undergoes rapid multiplications and it is produced many cells. So that's why what happened? Make the sea appears in red in color. So that is known as actually redtides or Mediterranean Sea. It is known as redtides or Mediterranean Sea. So these Goniaulux, they are released on toxic substance. The toxic substance is known as actually Saxi substance. What is called? Saxi substance. Now it is very poisonous. So that's why what happened? Many aquatic organisms inside of the water is there now. Inside of the water, there are so many organisms or it can survive, like fishes and some other organism. It will be die. Why? Because they die because of this dinoflagellates. They are released one toxic substance. So that's why there are many fishes. They will be die. Now discuss the euglenoids. So what about actually euglenoids? Euglenoids are mostly present in actually freshwater and marine. Euglenoids are mostly present in actually freshwater and stagnant water. Stagnant water means they are actually storage water. So where there water or storage, so in that areas the euglenoids are easily it can growth. So that is known as actually stagnant water. So euglenoids are mostly growing actually freshwater and stagnant water. This euglenoid, it is consists of a cell wall. That cell wall are generally enriched with actually protein layer. Means euglenoid, it is consists of actually cell wall. That cell wall is enriched with the protein layer. That protein layer is known as actually, that is known as, means such code is known as actually pellicle. Such is known as pellicle. So now what is the main functions of the pellicle? Pellicle main functions is, it can make their body is flexible. So that's why what happened, this euglenoid, it can move freely in anywhere. So that is euglenoid. Next, this euglenoid, it is consists of actually two flagellas. One flagella is long flagellas and another one flagella is very short flagellas. Euglenoid is consists of two types of flagella. One is long flagella, another one is very short flagellas. So anterior part of the cells, Euglenoids, anterior part of the cell, it is where the investigations of cytostome and cytopharynx and reservoir. So euglenoids are generally bearing actually anterior part. Anterior parts is bearing with actually cytopharynx, cytostomes and reservoir. So cytostomes means mouth part of the euglenoids, cytopharynx means gullet, and it is consists of another reservoir. So it is consists of a photosensitive stigmos or eye spot. Euglenoid, it is consists of a photosensitive stigmos and eye spots. So they are present in outer surface of the cell membrane. So they are present in outer membrane of the cell membrane or reservoir. Euglena reproductions. So what is the main reproduction in euglena? Euglena reproductions are mainly occurs through the longitudinal fusions or longitudinal binary fusions. What about actually longitudinal binary fusions? Both euglenoids are stand like these structures, and the binary fusions occurs in between two euglenoids. That is actually binary fusions. And polymellar stage of reproduction. So what is the polymellar stage? So polymellar stage means the group of cells or the group of spores are formed. So in euglenoids. So it is the reproduction part in euglenoids. It is the reproduction in euglenoids. And this euglenoid, so these euglenoids, most of the euglenoids, they are photosynthetic. Why? Because it is photosynthesis. Because of it is consists of a photosensitive stigmas. It is consists of a photosensitive stigmas. So that's why most of the euglenoids, they are photosynthetic in the presence of sunlight. Next thing. In deprivation of sunlight means absence of sunlight, these euglenoid act as predators or heterotrophic organisms. Absence of sunlight, this euglenoid act as heterotrophic organisms. At the same time, it is capture the small organisms, means it is absorb the, means it is uptake the small organisms. Like that, small organisms get uptake or absorbed. Okay, these pigments are identical to the higher plants. Euglenoids, which pigments are consist, those pigments are similar to the higher plants. So the best example of euglenoids is euglena is the best examples. Next one. So next we will discuss the another one is slime molds. What about actually slime molds? Slime molds generally they are saprophytic protistas. They are actually saprophytic protistas. What about actually saprophytic protistas? Saprophytic protista means they are absorb the food material from dead and decaying twigs. They are uptake the food material. They are uptake the nutrition from these dead bodies. That organisms is known as actually saprophyte. The body moves along with inglifying materials. The body moves decaying twigs along with the leaves or inglifying particles, such is known as actually plasmodium. Okay, the body moves along with the twigs and leaves of inglifying organic materials, such is known as actually plasmid. That is known as plasmodium. Such process is known as actually plasmodium. So in this plasmodium, under suitable condition, aggregations, it is produce a number of spores. And this plasmodium can easily grow for several feet. This plasmodium can easily grow and several feet. And it is bearing with actually spores of their tips. So it is bearing actually splits. Under unfavorable conditions, the plasmodium is differentiated forms of fruiting bodies and bearing actually spores at their tips. Under unfavorable conditions, the plasmodium is differentiated in that of indifferentiated. The plasmodium is differentiated in form of fruiting bodies and bearing spores. That plasmodium is bearing spores at the tips. Where the tips are there, the tips is consist of a spores. So the spores have a true walls. This spore is consist of actually true walls. The spore is consist of actually true walls. and highly resistant to survive for many years. So that's why what happened, it is highly resistant and survive for many years, and it survive for many years, and spore dispersals with help of airs. Spores dispersals with help of airs. Next, protozoans. So these protozoans I can classify into four types, that is amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. So what about actually amoeboid protozoans? The protozoans I can classify into four types, that is amoeboid protozoans, ciliated protozoans, and flagellated protozoans, and sporozoans. So now we are first going to discuss the amoeboid protozoans. So what about actually amoeboid protozoans? These amoeboid protozoans are mostly present in actually freshwater and as well in seawater, and most of they are present in actually moist soil. So they move and capture by the putting of food, means they move and capture by prey by putting by the pseudopodia. So what about actually these amoeboid protozoans capture the food material through the pseudopodia. Pseudopodia means, for example, it is amoeba. This amoeba cell wall, it is capture the nearby food material. Here, for example, here the cell wall is there. So this cell wall, the nearby cell wall, it is having actually small organisms are there. So this the cell wall is captured that organisms. Okay. So they move capture prey by the putting out by pseudopodia. It is capture the food material through the cell walls. That mechanism is known as actually pseudopodia. Example is amoeba. So marine forms have silica cells. So marine forms have, it is consist of a silica cells on their surface. So the best example is Entamoeba histalica. Some are parasites. The best example is Entamoeba histalica. Next, flagellated protozoans. So what about actually flagellated protozoans? They are free living organisms. So this flagella means this body is consist of a flagellas. They consist of a flagellas, and they are caused the diseases, sleeping sickness diseases. So they are actually parasitic. It is the cause the diseases is sleeping sickness diseases. The best example is Trypanosoma gramiansi. So this is the flagellated protozoans. Next one is ciliated protozoans. So what about actually ciliated protozoans? Ciliated protozoans means it is very aquatic, means most of the ciliated, they are mostly present in actually water. So that's why we are saying that it is aquatic organisms, and it is active organisms. They are actively aquatic organisms. It is known as actively aquatic organisms. Why? Because it is called as actively active organisms. Why? Because it is called as actually because of this cilia. So means it is consist of a number of cilias. This protozoans is consist of a number of cilias. So that's why it is called as it is actively moving in water. They are actively moving in water. So they have a cavity. This ciliated protozoan, it is consist of a cavity and it is open to the outer side. So it is consist of a cavity. That cavity is open to the outer side of the surface. Means it is open to the outer side. So due to the movement of cilia and water enters to the gullet. In this ciliated protozoans, with help of cilia, water and food materials are enter into the gullet. Example is Paramecium. So it is ciliated protozoans. Next, sporozoans. So what about actually sporozoans? They have an infectious spore-like stages like in their life cycles. In this sporophyte, it is consist of a spore-like stages. It is consist of actually spore-like stages. Okay. And example is Plasmodium or malaria parasites. Its caused diseases is malaria diseases. Its caused diseases is actually malaria diseases. So finished. Thank you. Thank you one and all watching of this video.